cyclic AMP or cyclic GMP. The pathways are very similar. And once again, cyclic AMP or cyclic GMP. It's a nucleotide. Adenine or guanine, the 5-carbon sugar ribose in a single phosphate. MP, monophosphate. But the cyclic versions, that phosphate is twisted around and one of the oxygens is rebound to the sugar. So the phosphate's twisted around in a circle. It's an unstable molecule. Once it's produced, it spontaneously hydrolyzes in fairly short order. So once you make cyclic nucleotides, they don't last very long. Stable? Yep. Okay, so we start out here. We have the receptor and a ligand binding to it. Whatever that, there are many different ligands and many different receptors that use this pathway. Question back there. Oh, ligand. In other words, the molecule L. The molecule that binds to that receptor. When that binding occurs, we now get a structural change in the receptor, which I've just drawn here. Meanwhile, we've got a trimeric G protein. Here's our alpha subunit. It's got a GDP bound to it. Once the receptor is bound to ligand, see this shape here? It means it can bind our trimeric G protein. And it's going to stimulate the GDP for GTP exchange. So now, GDP goes out, and GTP comes in. Recall, the receptor is acting as a guanine nucleotide exchange factor. In other words, it's stimulating the activation of this trimeric G protein. When that process occurs, the alpha subunit now falls off the beta and gamma. We'll put the beta and gamma right down here. And the whole assembly falls off the receptor now. Once we get that structural change in the trimeric G protein, the G protein falls apart and pops off the receptor. Now a second one could bind. So now we have our free alpha subunit, which is active. What it does is it's going to go and bind to and regulate a target protein, a membrane-bound enzyme. That enzyme is known as either adenylate cyclase, which makes cyclic AMP, or guanylate cyclase, which makes cyclic GMP. We're just going to, and we'll just look at cyclic AMP, but we'll say adenylate or guanylate cyclase. It's an enzyme that's embedded in the membrane. Now, of course, adenylate and guanylate cyclase are very similar to each other. It's just one produces cyclic AMP, the other produces cyclic GMP. So now what we see, we have our receptor, we have our intermediary, and now this is the start of the amplification steps. We're going to focus on cyclic AMP, cyclic GMP is very similar. What do you have to do to make cyclic AMP? You take an ATP, or GTP, you have to hydrolyze the second and third phosphates off to provide the energy to do this reaction here. So we start out with ATP. We convert it to cyclic, that's the small c, cyclic AMP, and then the two phosphates are covalently bound together called pyrophosphate if you're interested. But you have to tear those two phosphates off because you have to do that condensation reaction where you attach the oxygen of that phosphate back to the sugar again, and you have to bend that phosphate at angles that chemical bonds are not supposed to be bent. So the combination, the condensation, the bending of that phosphate, that takes so much energy, you got to knock two phosphates off of ATP to power that reaction. So now we got cyclic AMP or cyclic GMP. This enzyme will produce a lot of cyclic AMP molecules, so that's amplification here. So you turn one of these enzymes on, you may get 1,000 cyclic AMPs or cyclic GMPs. And what do these guys do? they bind to and regulate membrane-bound protein kinases. So we stick one right here, cyclic AMP. In this case, we're activating protein kinase A's, or PKAs. That's activated by cyclic AMP. There are protein kinase G's, a similar family that's activated by cyclic GMP. We're just looking at PKAs. Now, this is not just one particular protein kinase, it's a family of them. They all are activated by cyclic AMP, and they all have different target proteins when they're activated. So, when you turn these guys on, they phosphorylate lots of target proteins. So we've got two amplification steps here. The production of the cyclic nucleotides, each one of them turns on a PKA or PKG, and each one of those active kinases can now phosphorylate a whole ton of proteins. So we got preamp and a power amp here at the molecular level. So you turn one of these guys on and you ultimately affect hundreds or thousands of molecules in the cell. Now what we can get here, target proteins can include things like membrane transport, they can affect the cytoskeleton in some cases. They can alter transcription factors that regulate genes. They can alter cellular metabolism by ramping up or ramping down various enzymes and metabolic pathways. So when we turn these guys on, they have a wide range of targets depending on what PKA or what PKJ you use. And sometimes the targets are going to regulate other signaling pathways, ramping them up or ramping them down. So that's one example of a G protein linked signaling pathway. Now we have about 10 minutes left, and that is not enough time to cover the phosphoinositide pathway. So we will leave it here at this point, and we'll cover that coming in on Tuesday. So I'll see some of you guys in the lab.